Right, so welcome to this virtual poster presentation on uncovering human protein histidine methylation. I am Magnus Jacobsson, an associate professor at the Department of Immunotechnology at Lund University. And methylation of histidine is an elusive PTM that was discovered in the late 1960s, and it has only recently gained significant attention through the discovery of 3 human histidine methyltransferase enzymes. Corresponding to the SETI3 enzyme, the methyl-18 enzyme, and the methyl-9 enzyme. And the study that I'm presenting today represents the first system-level analysis of the PTM. And there are no affinity agents available to enrich histidine methylated peptides for proteomics. So to study the PTM, we therefore turn to an alternative approach. And we queried the most comprehensive DDA proteomics datasets available for the different flavors of methylation. And this allowed the generation of a protein methylome resource that could be explored using various informatic tools. And in total, we found over 2,500 methylation sites in HeLa cells. 300 of these correspond to histidine methylation, and almost all were novel. We also explored some fundamental features of methylation, such as the number of sites for proteins, the abundance of proteins carrying the different flavors of methylation, as well as the subcellular localization of the, the different methylated proteins. And we found that the histidine methylated proteins localized to all major cellular compartments in line with results on lysine methylation and arginine methylation. All right, we next explored the sites and domains targeted by histidine methylation, and we found histidine methylation to be overrepresented in several specific domains, and in particularly in zinc binding proteins. And concerning primary sequence, we found histidine methylation to be overrepresented in monospaced histidine-rich motifs, which is the region in these zinc binding proteins that coordinates the zinc atom. Finally, we explored the crosstalk of methylation and phosphorylation, and we found that the methylation events co-localize with phosphorylations that are predicted to be functionally relevant. In particular, one HME site localized in an ATP sensing loop in actin that is well conserved in evolution and is also extensively post-translationally modified, giving the opportunity for interplay between PTMs. So in conclusion, we showed that the human protein histidine methylation is abundant and widespread across subcellular structures. It's enriched in repetitive histidine-rich motifs present in zinc binding proteins.